Hi guys, my name is Ene-braye. Welcome to my channel, my YouTube channel, Nursing Digest with Joy, and we are going to be having a subject called microbiology and parasitology. Under it, we are going to talk about microbiology, introduction to microbiology. What we are even talking more is about the transient, the flora. Mostly we are going to be hitting more points on the floras today. So before we go into that, please like, share, subscribe. I don't know how to put it again. Like, share, and subscribe to my YouTube channel. That's the only thing you can do in blessing me. Okay, let's go straight. When you hear about microbiology, what comes into your mind? Microbiology, in the sense, in a layman's thought, is the study of microorganisms. Microorganisms are organisms that we cannot see with our naked eyes, except with the aid of an electron microscope or a light microscope or even a microscope. You can just put it in a microscope right there. So I say microbiology studies microscopic organisms, which I just said earlier on. When you hear micro, small, macro, big. So microbiology cannot study macroorganisms except it would have been called macrobiology. So we have the historical context. We have Anton van Leeuwenhoek was from 1632 to 1723, a Dutch scientist, not a Scottish scientist. Most people mistake it for a Scottish scientist while he was a Dutch scientist. He pioneered the microscope and described microorganisms as animalcules. He pioneered microscope and described the microorganisms as animalcules. So subdisciplines under microbiology, we have the bacteriology, we have the parasitology, we have more. Let me be listing them out: bacteriology, parasitology, mycology, virology, immunology. We will know that immunology is the study of immune responses. Bacteriology is the study of unicellular bacteria. Parasitology is the study of parasites. Parasites can be divided into two: the protozoa and the helminths. So the protozoa, they are unicellular, example malaria. Helminths, they are multicellular worms. And we also have the arthropods, they are vectors like mosquitoes, example ticks, mosquitoes. Those are the arthropods. So when we hear about mycology, what comes into your mind? Biology students, mycology is the study of fungi. When we hear of virology, what comes in your mind? Biology students, virology is the study of viruses. Do you get me? Good. So there was this thought that came up that living things emanated from non-living things. And when I was in my first year in nursing, I learned about abiogenesis. I learned about abiogenesis when I was in my first year in secondary school. I said first year in university. I learned about abiogenesis, and I knew a guy that his name started from Thomas, that he said that living things emanated from non-living things. So let's go. The person that stopped it. So when you hear of that living things emanated from non-living things, just know that they are heading to abiogenesis. So the key milestone here is Lewis Pasteur disproved spontaneous generation. So another name for living things emanated from non-living things is called spontaneous generation. Lewis Pasteur disproved spontaneous generation, proving microbes cause decay and diseases. So this Lewis Pasteur proved that microbes cause decay and disease. Why would you say life emanated from non-life? Sometimes they normally say, like, how do I put it right now? Like now, your food got rotten, and like maggot is feeding on it, or fly came to perch on it. You tell me that that fly is inside the food, or that fly grew out from the food, or the maggot grew out from the food. No, the maggot didn't grow out from the food. The fly never grew out from the food. So we're not talking about that today, but just when you hear spontaneous generation, just talk about abiogenesis, because proving microbes cause decay and diseases. Because you can't see a food that is infected with what's his name, maggots, and you eat that food. It will give you decay, it will give you disease. So in my next video, I'll be dropping about that. I'll be dropping about abiogenesis, because it's not abiogenesis we're talking about today, and how they disproved it. Thank you so much. So now let's talk about host-parasite relationship. What do you understand by host-parasite relationship? So let's look at the definitions. Hosts are organisms harboring another. They are also providing nourishment to them. So like now in humans harbor malaria parasites. Those are parasites called plasmodium. It's not so. So parasites, organisms living in a host, and some of them also causing harm in a host. When it comes to vectors, they are transmitters of diseases. Vectors are transmitters of diseases. Example, mosquito. Mosquito can transmit malaria, right? So we have vector types. We have the biological vector and the mechanical vector. Biological vector, pathogen undergoes life cycle within vector. Example, mosquito for malaria. Mechanical vector, passively transmit pathogens without life cycle. Example, housefly carrying bacteria. So this biological vector that is talking about pathogen undergoing life cycle within vector. So mosquito for malaria, that's mean it's the pathogen will undergo its life cycle within the mosquito. You understand? Before transmission. But this one passively transmits without life cycle. That's mean because this thing, house flies, you know that when they pick cholera, they go and, how do I say it? They use their mouth to touch a feces that has cholera, or a vomit that has cholera, or even a stool that has dysentery, and they go and pet someone's food. They passively transmit it, not that the pathogen grows in their body. But vector like mosquito, they are also vectors. Mechanical vectors, they are called mechanical vectors, but mosquitoes are called biological vectors, because that pathogen, it lives there for their life cycle. Like for it to develop, the plasmodium will not develop before they will pass it to a human. Do you understand? Yes. So let's continue. Host parasite dynamics. What do we talk about host parasite dynamic? We have definitive host and intermediate host. Let's look at them. Definitive hosts, they harbor adults, reproductive stage of parasite. Intermediate host harbors larva or developmental stage. Do you understand? So definite hosts, they are like humans for plasmodium. They harbor the adult or reproductive stage of that parasite. Mind you, these biological vectors we just talked about, like mosquitoes, the plasmodium, they are only in their body for their larval or their developmental stage. That means they have completed their larval stage before passing it. You, the human, now will be the one to, because mosquito, like malaria can stay in your body for months and you don't know before it shows up itself. So now it has harbored in the larval stage, in the larval life cycle. Then it's now passing it on to you so that you can harbor it for adult stage. So that's what we call about intermediate and definite host. That's host parasite dynamics. So we are done with that. Let's go to symbiotic relationships. So you know about symbiotic relationships, the ones that can harm, between parasites and hosts, the ones that can harm, the ones that do not harm. So we have 3 here on our list. We have mutualism, we have commensalism, and we have parasitism. Mutualism, the ones they both benefit. So I was talking about when the organism benefits and the host is unharmed, it is called commensalism. Example, skin bacteria. The host is unharmed and the organism benefits. So when we talk of parasitism, you know what parasitism is. You will not tell me your secondary school they never taught you about parasitism. Parasitism, their parasites benefit, hosts are harmed. Parasite benefit, hosts are harmed. Like worm in your stomach. When you did not see food inside the stomach, what does it do? So now when we talk about normal microbial flora, this is a place I used to normally enjoy. I like this place very well. So let me give you an origin, a hint. They said here that humans are sterile in utero. Colonization begins at birth via environmental exposure. Inside the stomach, you were sterile. I was sterile. Because inside the womb is sterile. We were all sterile. But when we came out, like when our water broke, we came out. Colonization began. The exposure of flora started. So we have types of floras. We have resident flora and transient flora. Resident flora permanently colonize sites. Example, Staphylococcus epidermis on skin. So we have Staphylococcus epidermis on our skin. And transient floras, they temporarily present, fail to establish. They can be in the body for like 1 to 2 months, then they die off and all the rest. So factors influencing floral composition: anatomic site, moist versus dry area. Example, mouth versus skin. The flora in your mouth is different from the flora in your skin. The flora in your skin is different from the flora in your anus, and so on and so forth. So nutrients, lipids or fatty acids favor specific bacteria. Do you understand that? So lipids or fatty acids, they favor some bacteria. Example, Propionibacterium in hair follicles. So pH, the vaginal flora has a pH of 4.0 to 5.0, dominated by lactobacilli. Infant gut pH differs by diet. Breast milk has a pH of 5.0 to 5.5. So age, those are still under the factors influencing floral composition. Age, diet, and health. Breastfed infants host more bifidobacterium. Cow's milk promotes adult-like flora. So now we want to enter into, we are done with factors influencing floral composition. I hope you enjoyed those parts. So opportunistic infections. What are opportunistic infections? Let's look at it. Normal flora causes disease if host is immunocompromised or flora relocates. Like if the flora relocates or the flora is no more there, we call it that the host is immunocompromised. And that flora, that normal flora, can cause disease in that particular area. Like now, E. coli going to the vagina where there is lactobacilli there and erupting. That's why most ladies, you see, sometimes they have this E. coli bacteria. E. coli is not in the vagina. E. coli is not in the vagina, hear me. It's in the anus. So as a lady, you are supposed to wash from front to back. not from back to front. Some of these ladies, when they have intercourse or sex, most of them, maybe they have their sex through anal, and this male organ entering into the anus will carry some fecal deposits in. Sometimes they will now go back to the vagina and have sex through the vagina. Some of them will carry these fecal deposits into, it's likely to happen, it must happen, into the vagina. So the vagina now will now be erupted with those fecal deposits, which the anus, normally the bacteria of the anus is called E. coli. So it will take it to the vagina, and the lactobacilli there will be erupted, and that flora relocates. And when the flora relocates, like the E. coli relocates to the vagina, it then causes E. coli infection. Not that E. coli infection is from toilet. Sometimes it can be from toilets or all the rest, but this thing normally happens when you wash from back to front. You understand? So let's go forward. So we have bacterial structure and classification. Let's look about the basics. Unicellular, prokaryotic. Their size is 0.2 to 1.5 micrometer wide, and they have 1 to 5 micrometer long. So the morphological classification is cocci, bacilli, spirilla, mycoplasma. This mycoplasma, they lack cell wall and pleomorphic. So cocci is spherical, bacilli is rod shaped, spiralis is spiral. So bacterial structure. Bacteria has external and internal structure. So let's look at the external structure. They have a capsule, that's their slim layer. They have cell wall. Their cell wall has peptidoglycan. Their membrane is made up of phospholipid bilayer. Their flagella is for motility. Their pili is for attachment. Then their internal structure has cytoplasm, ribosomes, which are for protein synthesis. So we are almost coming to the end of today's class, like today's section, because next I'll be talking about still bacterial growth and control. So their cell functions. Most of you know what cell wall functions are. Cell wall, they maintain shape, osmotic protection, enables binary fission. They protect the cell, like what goes in and what goes out, the osmotic protection, their shape and everything. So we are done for this class today. Please like, subscribe and share to my YouTube channel, Nursing Digest with Joy. Next time we'll be talking about bacteria and growth. Love you guys and God bless you. Thank you so much.